So there'll be a total of 4 questions and there'll be a directed team. The directed team will be chosen through a draw of the chit. The judges will be choosing the chit, and based on that, the orientation of teams will be decided. The directed team will get to choose the question, which will be displayed on the screen. After that, we'll have a 15 minutes timer, which will be your thinking time. During this time, all the teams can work on your solution. You will not be allowed to present anything on the board during this time duration. After the 15 minutes are over, the directed team will have to come on the board and present their solution. They'll get exactly 5 minutes to do so. After that, we'll move on to the next team. They'll get 3 minutes. The subsequent team will get 2 minutes, and the team after that will also get 2 minutes. The teams will get exactly 3, 2, and 2 minutes. There'll be no thinking time in between. After one question has been presented by all the teams, the judges will be asking for clarifications and questions. And after that, a member of the question making team will be presenting the solution on the board. At max, the directed team can get a total of 15 marks for if they answer the question perfectly. The next pass team can get a total of 13 marks. Then the subsequent team can get a total of 11 marks. And then the final team can get a total of 10 marks. I would also like to advise all the participants that do not repeat the exact solution of the previous. Sorry for the inconvenience. And also, I would like to just clarify that do not repeat the exact solutions of the previous team. Try to add new information to the question. If you repeat the exact solutions of the previous teams, that will not be considered for evaluation. If you have any further doubts, you can clarify them now regarding the format. Otherwise, I'll hand it over to the podium coordinators. Yes.
Suppose the directed team comes and could not finish the solution, can the next team continue from that solution? I mean,
Yes, you can continue, but yeah, you have to add on. Yeah, you can continue. There is no
I mean,
issues in that.
I can assume the facts that have been.
You can assume, but you can do that. There is no issues in that. Any other doubts? Okay, so I'll hand it over to Mrudul and Pranshu, the podium coordinators for the biology deep thought. All the best, guys.
Good afternoon. I welcome everyone to the Biology Deep Thought of Mimamsa 2025. My name is Mrudul.
And my name is Pranshu. We are the podium coordinators for Deep Thought Biology.
We'd first like to introduce our judges. We have Professor Satyajit Rath and Dr. Sarita Puri. Professor Rath is an immunologist. His work has examined mechanisms involved in the generation and activation of cells of the immune system.
Dr. Puri is a structural biologist. She works on protein misfolding and aggregation. We thank both of them for being here today with us. We request our judges to pick a chit to determine the team that gets to answer a question first, and we'll proceed clockwise from them.
So the order is IISc, IIT Bombay, IIT Delhi, and IISER Kolkata. Team IISc, you will be going first. We will now begin with deep thought biology. Presented on the screen are the 4 questions for this round. First question is, the memory remains.
Second question is, my heart will go on.
Third question is, grow a spine.
Fourth question is, the infection might be chronic, but this mutant is ironic.
I'll go with the fourth, I believe.
The infection might be chronic, but this mutant is ironic. One second. Microbiologist Ameya was studying Corynebacter goofus, a bacterium known to cause persistent infections in humans. Nihes and uricine are 2 factors released by the bacterium that contribute to its virulence, that is, the ability to cause damage to a host. Nihes is an enzyme that Corynebacter uses to break down proteins in its environment, from which it can derive essential nutrients. The NeHA gene codes for synthesis of Nihes. Uricine is a molecule used by Corynebacter for iron uptake under iron-limiting conditions. It is released by the bacterium into its surroundings, where it binds to iron with high affinity to form a complex. This complex is then recognized by outer membrane receptors, which facilitate its transport into the cell. When analyzing patient samples, 2 mutant strains of Corynebacter were found. NeHA1 mutants cannot produce Nihes, and OR1 mutants cannot produce uricine. Note, iron is essential for growth and survival of the bacteria. Ameya decided to study these mutants. He prepares mixed cultures of the strains and observes how growth patterns differ in different growth media. All cultures are sufficiently supplied with proteins, while the supplied iron is varied. The observations are graphed further ahead. In cultures 1 and 3, the initial population consists of the wild normal type, indicated by WT, and the NeHA1 mutant in a ratio of 9:1. While in cultures 2 and 4, the initial population consists of the wild type, NeHA1 mutant, and OR1 mutant in a 8:1:1 ratio. Note that cultures are well mixed homogeneous environments. The stacked bar plots show the relative frequencies of each strain at a given time, while the black dots are indicative of the total population density. Question 1. Based on the data provided, provide a mechanistic explanation for the differences in the growth of different strains in the mixed cultures. A drastic decline in the total population is observed in cultures 1, 2, and 3. Why is the same not observed in 4? Ignore effects of any new mutations that may arise over the course of the experiment. It is known that the production of Nihes is regulated by the given system. Ana R produces the energetically cheap and fairly short-lived HRN, which can passively diffuse in and out of the cell based on the concentration gradient. Above a set concentration inside the cell threshold, HRN activates Ana S, which further initiates the production of more Ana R as well as Nihes, which is transported out of the cell to carry out its own function. NeHA1 mutants, where NeHA is deleted, have non-functional Ana S. When artificial HRN is added to the setups 1 and 2, the NeHA1 mutants entirely dominate the population, reach 100% fixation. Question 2. Using the Nihes regulatory system, explain why the NeHA1 strain does not reach 100% fixation in 1 and 3. Question 3. The growth rate of a strain increases with its fitness. Construct a simple mathematical model deriving the fitness W of each of the strains and the average fitness of the population. The expression for fitness is of the form W is equal to baseline fitness plus net benefit from the molecules minus net cost of molecules. Subpart 1. How does your model explain the dynamics observed in the mixed cultures? Subpart 2. How have you accounted for the Nihes regulatory system in your model? Assume an infinite population. The baseline fitness can be assumed to be the same for all strains, while the net benefit and net costs are functions of the population composition as well as the individual benefit/cost of each molecule. The individual benefits provided by Nihes and uricine can be assumed to be B1 and B2, while the individual costs to produce them can be taken as C1 and C2 respectively. Your model is expected to give fitness of each strain as a function of B1, B2, C1, C2, and the frequencies of each strain. IISc, do you need any clarification? If you don't have any clarification, teams, you have 15 minutes of thinking time, and your time starts. Teams, you have 5 minutes remaining. Teams, you have 2 minutes remaining. Teams, you have 1 minute remaining. Team, your time is up. IISc, kindly come up to the stage to present your answer. You have 5 minutes to present your answer and your time starts now.
Okay, so I would like to start by explaining the overall scheme. So, could I go to the diagram? That would be helpful over there to explain. I'd have to draw otherwise. Yeah. So in scenarios 1 and 3, where the only difference is iron concentration in media, and the changes in iron concentration only matter for the regulation of URV gene, which of the urvicin, of the expression of urvicin, which is same for both NEH1 and WT variants. So we essentially see no difference in the graphs. Now, why we see a decline of overall population comes from the relative fitness of these two variants. Fitness, as described here, is the baseline fitness plus the benefit minus the cost. In case of the wild type, we have both of these factors. Now, if we were growing the wild type and the URV, in case we were growing the wild type and the NEH1 separately, NEH1 would obviously not survive because of a lack of the entire HRN pathway. However, when grown together, wild type and in this case, NEH benefits from the molecules, since the molecules are released into the medium and can be passively transported through the membranes. The molecules released by an initially high concentration of the wild type are used by, the benefits of those molecules are reaped by the NEH1 variant. So they do get the benefit, but they do not have this extra cost, in effect raising their overall fitness in the mixed medium. Hence their proportion rises. However, due to a decreasing proportion of the wild type mutant, their contribution of HRN to the system decreases, leading to an overall decrease in population that we see in both those graphs. In graphs 1 and 3. In graph 2, the URV1 variant and the wild type are essentially similar because because of high iron concentrations. There are no practical differences. Their relative concentrations doesn't change, and it is mostly what happens in graph 1 with respect to NEH1 and the wild type. For graph 4, similarly, the benefits are reaped. The benefits of the molecule urvicin are being reaped by URV1 that are produced by the other two strains, and it is an increase in their population that corresponds to a overall stable net population. Now, it could be possible that the urvicin complex is not, the entire complex might not be transported through the membrane, maybe just the ions are, because it doesn't state that explicitly in the question. I'll go with that assumption. So because iron ions are being transported across the membranes and urvicin molecules are present in the medium, those molecules can be harvested for a long time, thereby keeping a stable population size despite a decrease in the populations of wild type and the other one. Question?
If we assume the frequencies to be F1 and F and baseline fitness to be K, then the fitness of each strain will be given as K plus F1 B1 plus F2 B2, as from the formula you can see minus F1 C1 minus F2 C2.
NEH1 does not reach 100% fixation because they still need the molecules being produced by the wild type. If the wild type goes out of the population, the entire population collapses. which is not the case in case we provide HRN from into the medium synthetically.
I see your time is up. IIT Bombay, you have 3 minutes to present your answer, and your time starts now.
Yeah. Sorry. So for the first graph, we can see that the initial population, we have a higher percentage of wild type population. So wild type population would like corresponding to this equation which they have written, the NEH1 is not able to produce NEH gene, so the cost of it would be lesser, but the wild type is still bearing the cost for NEH production. And while the benefits both, because the receptors of on the outer membrane, they are not disappearing in this. Only the gene for production is disappearing, which gives the wild type and the NEH1, both of them, they are like able to reap the benefits of NEH1. So hence the total, like this W factor fitness is more for an NEH1 as compared to wild type and iron rich and both iron poor media. But later on, like as days pass, the wild type decreases, which causes lesser production of NEHs, which corresponds to an overall decrease in population. For the iron rich media graph 2, it is similar because Orvicin is like not really effective when we have iron rich media. But for the fourth graph, when we have an iron poor media, then Orvicin 1 is being produced by both wild type and NEH1, which has a cost on them, but URV1, it does not have any cost for producing Orvicin, but it benefit equally as the two. It causes a high increase in population of URV1, and why the total population is not decreasing is because when Orvicin goes out and it binds with Fe3+, it causes Fe3+ to just go inside, and the Orvicin remains outside, which does not like limit the supply of Orvicin, which sustains the population of URV1. And these are the equations for the third question, where B1, B2, C1, C2, B1 and C1 are the benefit and cost.
Bombay, your time is up. IIT Delhi, you have 2 minutes to present your answer, and your time starts now.
So in the first graph, the enzyme is only produced by, so due to which W produces NH, and NH1 does not produce any NH, due to which the enzyme released by WT will also be used by NEH1 as well as the rest of the WT, due to which the population of the WT will keep decreasing, which is indicated by the graph, and NEH1 will keep increasing till some certain value, due to which as the ratio is 9:1, the decrease will be quite rapid, whereas the increase will not be that rapid, due to which the total decrease is indicated by this graph. For the second one, it's quite similar to the first one. That is due to because Orvicin also
Yeah, so I considered, this is the equation we get from while expressing the
Delhi, you have 30
W.
seconds.
Yeah. So when we put in the concentration factors for each of them, we see that the net delta from this term and net delta from this term reflects the way the change in the bar plot.
Okay. The third graph is similar to the first one, as it does not depend on URV one, UV seen released. That's why
Delhi,
the
your time
first
is
and the
up.
third graph is
IISER
same.
Kolkata, you have 2 minutes to present your answer, and your time starts now.
So basically what happens is that our immune system deprives the environment of these essential iron and micronutrients which can be used by these bacteria to grow. So these bacteria develop systems like NAHase and uracil to catch up the iron. So NAHase is basically a protease. It breaks down proteins, and proteins contain iron as cofactor. And uracil is responsible for getting the iron to the membrane of the bacteria. So these two, both of these pathways can be set to like enrich iron to the bacterial cell. So what happens here is that we must see that in a consortia where we are having 2-3 microbes competing, it all thing matters is the cost, like how much a bacteria is spending, how much a bacteria is getting. So the expenditure here is like very much considered, because both of the things which are released are being released in the extracellular environment, and in the extracellular environment that particular thing can be used by all of those bacteria which are secreting and which are not secreting. So this would be the reason that would explain that why the population is not declining in the fourth one, where we can see that would be seen is like we can see that the bacteria have somehow managed to grow together in harmony in the fourth. And for the second question, it's a typical circuit of a Coren sensing system where the bacterial population is communicated through this actively diffusing molecule known as HRN.
Kolkata, you have 30 seconds.
So what happens is that as the HRN concentration goes up, it signifies the bacterial population. So when we can see that in third scenario, the bacterial population is the density is very low, so it is not able to produce that much of HRN, and hence that circuit is not activated. So it can be explained through the analogy of an Coren sensing system where
IISER Kolkata, your time is up. And now the floor is open for questions from the judges. Judges, any questions?
So, since there was a lot of talk, I may have missed this. In graph 3, where there is no iron limitation, all 3 strains have been added. Yet, only one strain is dominant. Is there an explanation that anybody provided or would like to add?
I had a slight clarification. I think you mean graph 2. Graph 3 has a iron depletion and just 2 strains.
sorry, graph 2.
Yeah, well, so in graph 2, the wild type and URV1 show no differences in their survivability, because it's a high iron medium, and the difference that URV makes only kicks in low iron conditions. So that is why there is no differences between wild type and URV, whereas NEH1 in this case is the only one which gets the benefit of using the molecules being secreted by the other 2 strains, namely HRN, where while not bearing the cost for it. So fitness only goes up for NEH1 in this case, not the other 2.
Why type competing you are V1?
It,
Is not outcompeted.
there isn't no. The wild type isn't, right? But wild type is more or less the same. If anything, URV is increasing a bit.
And now we request a member from the question making team to present the correct answer on stage.
I'll go to the first question. Am I audible to everyone? Okay. First slide. Okay, so you have two mutants here that are not producing one kind of molecule. The Na1 mutant isn't producing NAHES, which is an enzyme that the bacterium secretes out of the cell and degrades proteins in its environment, and the degraded product is used to can be used as a nutrient. UR1 cannot produce orvicine, and orvicine is a mutant that goes out of the cell and binds to iron, and the iron orvicine complex is the only thing that the bacterium can take in. Both these molecules, since they can be released by one bacterium and can be used by the other, anyone else in that population can be called as a public good over here. So this system is a typical example of something called a public goods dilemma, where you have one or more public goods that benefits everyone in a population equally, regardless of how much they contribute to producing that good. So over here you have, in any case, it's called a dilemma because your benefit, you get a benefit regardless of how you contributed to it. You are incentivized to cheat and freeload off the people who are the individuals that are producing the molecule. So over here you have two cheaters in the population that are the Na1 mutants that is cheating for NAHES, and the UR1 mutant that is cheating for orvicine. So in graph 1, since you have added iron-rich media and you have added the wild type and the NAHES in a 9:1 ratio, the NAHES mutant does not have to pay the cost for producing NAHES, and therefore it has a fitness advantage against the wild type, and therefore it dominates the population. The population collapse you see is because you reach a point where there are no individuals that produce NAHES anymore, so there is no food for anyone in the population, and the public good has collapsed. Similar explanations for 2 and 3. In 2, even on adding the orvicine, like you said, it doesn't make any difference because iron is not limiting factor here. What happens in graph 4 is now you have two cheaters in the population, and now one cheater, now of course both of them have a relative fitness advantage against the wild type because both of them have lesser costs. But now the two mutants are forced to cooperate because each of them have to cheat on the other for the molecule that they are not producing. So what you can infer from 4, the reason that why orvicine dominates, they're both cheating on each other, and they could have formed like a 50-50 population, the two mutants, but clearly orvicine dominates. And from this you can infer that the cost of producing orvicine is far more than the cost of producing NAHES. So there's an asymmetry in the cost of producing these two molecules, and that's why the orvicine mutant has to produce that much less of a pay that much less of a cost, and again gains that much of a fitness advantage. Population does not collapse because also you see a bit of wild type and NAHES left, because that tells you that the population can survive on this much amount of orvicine in the molecule, and that can sustain the whole population. It doesn't collapse because there is also enough people producing NAHES, which you need far more of. Next question. Next question. Yeah. So this is, as someone mentioned, this is a classic quorum sensing circuit. The NAR produces these energetically cheap molecules called HRN, which can diffuse passively in and out of the cell based on the concentration gradient. It would move out of the cell if you have more HRN inside. It would move inside the cell if you have more HRN outside. You can imagine that there'd be more HRN outside when you have enough cells in your environment producing HRN. So what HRN is, technically it is a check. It's a sensor for cell density. It since NIS has a threshold for HRN beyond which it activates, HRN is sort of regulates a behavior that only gets activated when there are enough individuals producing this molecule. And so NAHES production, what you have done with this circuit is link NAHES production to a density dependent phenomenon.
So you need enough HRN producing individuals in the population for NAHES to work. So the question is, in the graph 1 and 3 and even 2, you can extend this. Why does the NAHES mutant not reach complete fixation? In that, why is there a bit of wild type still left? The answer to that is, like I said, you still need HRN producing HRN producing individuals to survive in the population for NAHES production to be activated. At some point, as NAHES's frequency increases, since they are not producing HRN, the amount of HRN producing individuals goes to 0 or very low, and this shuts down the production of NAHES in all individuals. So now the benefit that anyone was getting from NAHES goes to 0 or very low, and so does the cost, which renders the wild type and the NAHES mutant effectively the same. Third question. Yes, a simple mathematical model, you have sort of got the idea, but you have to consider the fact that the benefit scales with the frequency of the individual in the population that's producing the molecules. So could I have a chalk? So for a wild type, we are expecting something like you have a baseline fitness, you have benefit you gain from NAHES, which will be the frequency of the NAHES producing molecules, which in this case is UR1 plus the frequency of wild type. Likewise, benefit you get from urvicine will depend on the frequency of the urvicine producing individuals in the population, which is the frequency of NAV1 and the frequency of wild type. The cost does not scale with frequency because your individual cost is not going to matter. It is not going to depend on how many individuals are producing it. So it is just C1 minus C2. For the mutant strain, the only thing that would change here is that for the NAHON mutant, this would be 0 because it's not paying the cost for it. And for the UR1 mutant, the C2 would go to 0. The last part was how do you incorporate the NAHES regulatory system's effect on in your model? I would have taken any threshold function that beyond a certain frequency of the NAHES individuals, that is the non-HRN producing individuals, meaning the others are so low that there's no HRN system anymore, the benefit and the cost go to 0. So just multiply this thing by something like a decreasing Hills function, and that would have been an answer. So something that goes like this: you have the frequency of the NAHES mutant and the function. It would go something like this, and this is the threshold. So anything like this would have been an answer. Thank you.
IIT Bombay, please choose from the remaining questions.
Grow was fine.
Grow with Pine. Ishan, IISER Pune's budding embryologist, has been studying the development of a vertebrate embryo. His work mainly focuses on the development of somites in this organism. Somites eventually form structures like vertebrae. Somites are cohesive groups of cells that are separated by spaces called clefts. The grouping of cells to form somites happens one after another, starting in the region of the head and ending in the tail. The image above shows you the arrangement of the tail bud and the presomatic mesoderm in the embryo of the vertebrate. Tail bud is the only region of active cell division, and cells are added to the PSM from the tail bud as shown by the white arrows. Somite formation is influenced by the opposing gradients of two molecules, RO and JEF. RO is a small molecule, not a protein, produced by the head region and newly formed somites. RO diffuses towards the tail. This sets up an RO gradient. A protein called JEF also has a gradient, but in the opposite direction. Only the cells in the tail bud can transcribe the gene encoding for JEF. Keep in mind the JEF protein has limited diffusion ability, which is not enough to establish a gradient on its own. No other cells or molecules are involved in the establishment of the JEF gradient. Question 1. How do you think the JEF gradient is being established here? How are the gradients of RO and Jeff maintained? Somite formation is also influenced by an intracellular protein called HOP in cells of the PSM. HOP is essential in somatogenesis. The HOP protein can directly inhibit the expression of the HOP gene. Essentially, it inhibits the transcription of its own gene with a delay. Question 2. Plot the concentration of HOP protein versus time in a single cell. In further studies that Ishan conducted, he observed ligands called kappa and their receptors called sigma on the external surface of all cells in the PSM. Binding of kappa to sigma activates sigma. Activated sigma increases the rate of transcription of the HOP gene, and the HOP protein that is formed inhibits kappa. Kappa on the surface of one cell cannot bind to sigma on the surface of the same cell. The time delay due to kappa sigma signaling is comparable to the time delay of the inhibitory feedback of HOP protein to HOP gene. Presomatic cells are cells of the PSM which have not formed somites. Refer to the chart and diagram below for further information. When differentiation happens, somite boundaries form somatic clefts. Question 3, subpart A. Draw the concentration of HOP protein versus time in the same plot for neighboring cells along the head to tail axis. Subpart B. What would you expect to happen in a mutant where the kappa gene is deleted? Question 4. Based on the information given in the question, qualitatively explain how somites are being formed. Use information from points B, C, and D and your answers to questions 1, 2, and 3A. Question 5. If the final size of the organism is constant, what parameters are deciding the number and size of somites? IIT Bombay, do you need any clarifications? If not, then your thinking time starts now. Teams, you have 5 minutes left. Teams, you have 2 minutes left. Teams, you have 1 minute left. Teams, your time is up. IIT Bombay, kindly come up to the stage and present your answer. You have 5 minutes and your time starts now.
Okay, so for the first question, it asks how is the Jeff gradient being established? So we are given that the tail bud is the only position where Jeff is transcribed. So when the cells divide in the tail bud, they will contain the transcription of Jeff and will also contain Jeff, and it will be added to the PSM, and these cells will then form the somites. So the top cell also would have some Jeff concentration because it is being produced from the tail bud, which then these cells go to the PSM and then form the somite. And also it's given that it diffuses, but the diffusion is limited. So as we come closer to the tail, the Jeff concentration will be more because the tail bud is the only position where Jeff is transcribed. Also, how is the RO and Jeff gradient maintained? So RO is being produced in the head, and so the concentration or near the head would be higher, and also the somites near the head would have spent more time as being a somite, so it would produce more RO. So as the somite is closer to the head, it will have more RO concentration, and it will decrease as we go down.
So for the second question, the HOPP concentration is approximately 0 if we take at time 0. It increases as the gene gets transcribed, and because it's the same molecule which is acting as the inhibitor, the concentration reaches a plateau after some time. The cells which are near the tail region have a higher concentration of HOPP, as we can see from the graph which is the chart which is given below. A high amount of HOPP is responsible for somatic body formation, and since the somatic cells are formed from the tail side, first the body gets formed and following that the boundary gets formed. So from here we can conclude that HOPP is higher towards the tail region and it decreases in concentration towards the head. So that's why this graph is formed. So then for the fourth question, how can we qualitatively explain how somites are being formed? So the similar explanation that near the tail region, the HOPP is in higher concentration, due to which it forms the body. And as the cells move upwards, the concentration decreases and the boundary gets formed, and the somites get separated from the PSM. And if the final size of the organism is constant, what is the difference in the number and size? So basically, if the size is constant, if the number of the somites are higher, the size will be smaller, and vice versa. So it's basically the HOPP concentration which is deciding this. So if the cells have a higher HOPP concentration, the body will be larger, so the size will be more and the number will be less. And if the concentration is lesser, the boundaries will get formed faster, so then the size will be smaller and the number will be more. Mutant wala.
In question 3b, basically kappa gene is in the mutant there is no kappa gene is deleted. Because of that, hop protein, hop gene won't be formed because there won't be any activated sigma, and because of that, hop protein won't be formed, and so there will be low hop protein concentration, which is present in somite boundary formation. The cells responsible for somite boundary, and therefore the boundary part will increase and the somite body part will decrease.
So in 3B, when there is less HOPP, there would be more boundary formation. So there would be like more of a ratio of boundary than cells, than the cell body. So there would be more boundaries, which would cause the cells to be smaller and hence more in number.
$30 left. IIT Delhi, you can now present your answer. You have 3 minutes and your time starts now.
So since the tailbud is the only region for the formation of JEF protein, the JEF gets continuously formed. This portion is the head. And as the molecules grow, the JEF can't transverse. So the JEF that was already produced here be here only, while the concentration for JEF will be maximum in the tailbud region since it is the site of active production, which creates a gradient in the opposite direction from tail to the head. And the as RO is a small molecule which could move, which could diffuse easily, and it is produced at the head. It diffuses downwards, which maintains its gradient in the direction from head to tail.
In question 2, initially, since there is a time delay of the inhibition of HOPP, due to its formation, its concentration increases, then it reaches a maximum, and because of the inhibition, it then starts to decrease again. And then when there comes a minimum, the inhibition is reduced so much that again the production of HOPP will dominate, and then it will increase, and thus the cycle will go like this. And in question 3a, since HOPP is produced from the PSM, there is a gradient from head to tail, and thus there is a slight shift in graph in the concentration of HOPP in the neighboring cells. So it would be something like this.
And now, the protein was responsible for the
30 seconds left.
somatogenesis process, which is essential for the somite formation process. Due to this periodicity, the formation of somites, which coincides with the maxima and minima of the graphs. These are the boundaries or the spaces near the minimas.
Your time is up. IISER Kolkata, you can now present your answer. You have 2 minutes and your time starts now.
Hello. So basically, somite development can be thought of as a signal, and it is regulated by an AND gate where two signals are required, JOF and RO. So when both of these signals are present simultaneously, then we can assume that a group of cell differentiates into somite, and this development takes place through the kappa sigma signaling, where this is type of an lateral inhibition, where you have some identical looking cells, but due to an external signal and stochasticity, this guy inhibits its kappa signal, so these two cells are separated, and this group of cells develop two somite cells, and these are pre-somite cells. And the hop can be thought of an negative autoregulation network motif, where hop itself is negatively regulating itself. So it's like first of all, its production increases with time, and then when after a certain threshold, when its concentration goes above, so that it can bind to its promoter and inhibit itself, it reaches a lower threshold. So it helps a very quick, like the time lag for the activation of hop is significantly reduced here. And if we consider the order of the development, we can see that infer from this graph that
30 seconds left.
we have an order where first the pre-somatic cells are there, from after that the kappa beta signaling takes place due to the AND gate which is acting. Then there is a somite boundary which is formed, and from that somite boundary, after the hop production increases, we have a complete somite body production.
I see you can now present your answer. You have 2 minutes and your time starts now.
Okay, so firstly, to give a better explanation of how somite boundary formation happens, draw Jeff first. As it was previously explained, Jeff gradient depends only on the amount of time, Jeff gradient at a particular spot on the body depends only on the time it has elapsed from that point being, well, the tail region. So with time, the Jeff would reach below a certain threshold, the threshold shown in the diagram, below which the cells transition from presomatic to either body or plate cells, boundary cells. So if it happens at such and such time where the concentration of Jeffs drop down, at that time, if this is the concentration of hop in a cell further away from the tail, and this is in a cell closer to the tail, the hop, as previously explained, reaches a maximum, then drops. At that moment, if hop concentrations are higher, the cell would form a body. If it's lower, it forms the boundary. And the number and size of boundary is controlled by the periodicity of this Jeff of hop concentration going up and down.
30 seconds left.
If it's a short period, whereas the relative concentrations in this table of the peak concentration of hop and its threshold concentration, if that ratio remains constant and the period becomes shorter, the number of boundaries versus bodies would the number of boundaries versus bodies will, both boundaries and bodies ka number will increase, whereas their relative lengths will remain the same.
Your time is up. The floor is now open for questions from the judges. We request a member from the question making team to present the correct answer on stage.
Put this in your pocket.
Hello, I'm audible.
Yes.
Hello. I'm audible, right? Yes. So I'll use this space. So as let this be a developing organism, and as we know, the cells are dividing in the tail region, and the organism is going in this direction, and the cells kind of move in this direction. So essentially, the further away the cell is along this line, the further like the more time it has spent away from the tail bud. And as we know, Jeff is being produced only in the tail bud, and the moment a cell leaves the tail bud, Jeff is no longer produced. So it starts getting decayed, like Jeff and its mRNA start to decay over time. So as a result, the further away you go from the tail bud, the more time the cell has spent away from the tail bud, therefore Jeff has decayed, and therefore you get a Jeff gradient in such a direction. RO on the other hand is being produced over here, and RO is diffusing in this direction, so we get a opposing RO gradient. These two gradients can very much establish themselves independently of each other, but one mechanism that could support this gradient formation could be either RO speeds up Jeff decay or Jeff speeds up RO decay by some kind of mutual inhibition, which kind of sharpens the gradients, so as to speak. So this is how the gradients could be maintained. And as for the hop part, so the hop protein is providing negative feedback upon itself, but the negative feedback comes with the explicit time delay. and that gives us oscillations. So like if we didn't have the time delay to just reach and like plateau off somewhere, but what this time delay does is that you reach a maxima, and the effect of the maxima is felt later, which gives us this minima, and the effect of the minima will give us a maxima again, and you get continuous oscillations of a specific time period. Now what this kappa lambda signaling would do, like as some people correctly identified, kappa lambda can lead to lateral inhibition, but there's a caveat here. The kappa lambda again comes with a delay of its own, which is comparable to this hop signaling the hop's delay upon itself. So what we essentially end up with is hop negatively feedbacking upon itself, and by the kappa lambda signaling, it is negative feedbacking, giving a negative feedback on its neighboring cells as well, with the same degree of time delay as itself. So what essentially what this leads to is that neighboring cells, their hop cycles sync up. Like if you were to draw graphs of two neighboring cells close by, they would be something like these. They would be nearly overlapping, and the phase difference between them will be negligible at best. So it can be assumed that the entire organism, like with minor phase differences here and there, it can be assumed that all the cells are nearly in the same phase. Like all cells simultaneously have high hop, low hop, high hop, low hop in an oscillating manner. And finally, can we move to the graph? Yeah. Yes. So finally, as the graph says, when Jeff is high, cells remain undifferentiated in the presomatic form, but when Jeff gets low, cells have to differentiate. In that case, if Hoff is high, you get a somite body. If Hoff is low, you get a boundary. So as the organism is going in this direction and the gradient moves this way, you kind of get a threshold. So like as long as cells are above this, in this lower region, and they're above the threshold, they remain undifferentiated. But as the gradient moves in this direction, eventually cells will start to differentiate and have to form somites. So since the entire thing is oscillating in a like high hop, low hop, so if the cell crosses the threshold while it is in a high hop state, then you get a somite with it expressing genes for somite body formation. And if it's crossing the threshold while it is in a low hop state, you end up getting genes for boundary formation. So essentially the PSM, like high hop, low hop, like high hop, low hop, and you end up with somite boundary, somite boundary, somite boundary, and hence the entire organism gets divided into somites. And finally, as to what regulates the size and number of these things, it will be the time period for Jeff degradation and Hop oscillations. If the time period for Hop oscillations were to be sped up, we will end up with much smaller somites and much larger number of somites. And likewise, if you speed up the like Jeff degradation, you will end up with much larger somites and much smaller numbers, and vice versa. Hope this helps. Thank you.
IIT Delhi, please choose one of the remaining questions.
The second one, my heart will go on.
My heart will go on. Mridul was studying heart muscles. He used an experimental technique where a live single cell could be imaged and free calcium 2+ buildup in the cytosol, that is the cytoplasm without organelles of the cell, could be visualized as fluorescence. These fluorescent visualizations are referred to as sparks. When a cardiac muscle cell is stimulated, calcium influx occurs into the cell through diffusion due to the opening of ion channels that only allow calcium 2+ to pass through. In an unstimulated single cardiac muscle cell, a calcium spark may appear suddenly in an otherwise normal cell. This is an extremely rare occurrence. Blocking of the calcium channels on cell membrane or membrane disruption does not affect the occurrence of sparks. When the unstimulated cell was injected with a drug called IISER, the frequency of calcium sparks increased. IISER affects the somicosome, a cell organelle in the muscle cells, by increasing the affinity of GNR, a receptor of calcium present on the surface of the somicosome, towards calcium. Question 1. What is the role of the somicosome in the cardiac muscle cell? Question 2. Why do we see sudden calcium sparks in an unstimulated cardiac cell? A GNR molecule has only one active site for calcium binding, which causes change in the conformation of GNR. A model was designed to account for the change in conformation of GNR due to calcium binding. Refer to the plot given ahead. The black line shows the theoretical curve which assumes that calcium ion influx into the cell through the voltage gated calcium 2+ channels is enough to cause appropriate change in GNR conformation. Red colored curve shows the experimentally measured value of the change in GNR conformation. Note that there are no intermediate signaling pathways between the effect of calcium on GNR and the effect of GNR on chromosomes. Question 3. What is happening at the level of the GNR receptor that is giving us the experimental curve, and is there any advantage to it? The adjacent diagram is a schematic representation of the conduction of electrical impulse in the heart through a region called the Shrishita. Path alpha containing alpha cells and path beta containing beta cells are the two paths that the impulse takes to travel from chamber 1 to chamber 2. Usually the path alpha and beta are identical in terms of impulse conduction. The impulse is like an electrical signal passing through a circuit. The impulses are not combined at X, and the impulse will be sent ahead of X if the impulse reaches X from either arm. X acts like an OR gate. In a certain mutant human, it was found that beta cells conduct impulses faster than alpha cells. Alpha and beta paths can conduct impulse bidirectionally. Cells after conducting an impulse enter a recovery phase during which they are incapable of conduction of another impulse. Alpha cells have a smaller recovery time than beta cells. Question 4. Despite this mutation, impulse conduction through the Shrishita occurs normally. Explain how this may happen. The electrical signals in the heart occur in a rhythmic fashion. This means after one impulse, there is some time period before another impulse arrives from chamber 1 at Shrishita. Question 5. What could be the possible outcome if a premature electrical signal, that is impulse coming in the time period between two regular signals, arrives at the Shrishita in such a mutant human? That's the question. IT Delhi, do you need any clarification? Teams, you have 15 minutes and your time starts now. Team J have 5 minutes remaining. Team J have 2 minutes remaining. Team J have 1 minute remaining. Teams, your time is up. IIT Delhi, kindly come up to the stage and present your answer. You have 5 minutes and your time starts now.
Yeah. So initially the Ca2 plus is freely flowing in the cytoplasm. But it will be get attracted towards the walls of the sumicosome, and it will just, the Ca2 plus ions will just bind with the GNR, due to which the concentration of free Ca2 plus ions in the cytoplasm will decrease, leading to the opening of ion channels, due to which the diffusion process will happen.
For the question 2, if the cardiac cell has not been stimulated for a long period of time, due to the continuous outflux of calcium 2 plus ions outside, there would be a high diffusion gradient between the inner and the outer surface of the cytosol, due to which there would be a sudden influx of calcium 2 plus ions, and which would be depicted as sparks.
So the explanation for 3 might be the fact that there is a presence of another molecule, say AX, where the presence of ions A stimulates stimulates the GNR receptors. So what happens when Ca2 plus is added to this system is that Ca2 plus has a lot of affinity for the ion X. So a complex CaX forms plus A ions. So this process happens readily. So the graph that is obtained experimentally is very similar to the one that is obtained during strong acid strong base neutralization.
I also wanted to add on the second part that the Ca2 plus will come near the boundaries of the sumicosome, due to which there will be Ca2 plus build up, and due to these sparks will be observed in an unsimulated card.
IISER Kolkata, you have 3 minutes to present your answer and your time starts now.
Hello. So there are 3 things in the picture. First, this is the organelle somicosome. This is the outer cellular thing, and this is the cytoplasm. So there are 2 like channels, we can say. First channel is on the membrane, and the second channel, which is GNR, it's on our organelle. So the first question is, what is the role of this organelle in cardiac muscle cell? So this particular organelle acts as a reservoir for calcium ions. So basically what happens is these muscle cells, these cardiac muscle cells need to respond to nervous stimulus to contract and expand, which is mediated through this calcium signaling. So there's a very high difference in concentration of calcium in the extracellular and the intracellular thing, which is mediated, which is like about 10 K power minus 3 molar something. So this acts as a reservoir for creating the difference in calcium signaling. And the second question is, why do we see sudden Ca2 plus sparks in unstimulated cardiac cells? So the probable reason could be these pumps which are present on the which is present on the organelles. So basically pumps open and close in a probabilistic way. So they are like fluctuating always, and these random fluctuations can lead to sometimes what we see fluorescence, which is free calcium, which is being released in the cytosol. The third thing, it's a graph. So basically, what we are seeing is that in this thing, the graph from a little bit sigmoidal to shifts to an hill function where it becomes more switch-like. So this shifting from more switch-like says that our cardiac muscle cells are like more developed to respond in a very quick way. So
and it is governed by the Michaelis-Menten equation according to dPdt, S for muscular S to the power n. For question number 4, despite the mutation, impulse conduction So when it is coming to this point, at the same time, alpha and beta converts faster, and this x will be
30 seconds left.
comes together, then only the beta one, alpha one is remaining over here, and then there is a time for recovery. Beta has a longer recovery time. So then any conduction comes from the alpha, so it is beta, alpha, and then again alpha has to recover, alpha cells have to recover. So again the next conduction happens to beta. So it is a subsequent one. What is the possible outcome if a premature electric signal arrives at the critical signal? When alpha and beta both.
Isaac, your time is up. IISc, you have 2 minutes to present your answer. And your time starts now.
Okay, so building on the previous one, because alpha has a alpha has a longer time of conduction and a shorter recovery time, and beta on the other hand has a shorter time of conduction and a longer recovery time. They would essentially, over a long period of time, they would form a sort of a what should I call it? The conduction alternates between the time of impulse aligns with these two in such a way that at any given moment either one of the two cells are ready to conduct the impulse forward, and impulse coming between its set times would come at a time where either one of the cells are either stimulated or in their recovery time, not which prevents the signal from propagating. About the chomicosome, the it's unstimulated.
To elaborate
30
more,
seconds left.
to elaborate more on the third part, on the Shomikosome one, obviously the probabilistic model is fair, but we thought it was more of a, when in a very high Ca2 plus concentration, the entire Shomikosome membrane becomes permeable, and that causes the cytosolic Ca2 plus to increase. The fraction of GNR whose constant conformation is remaining same implies there is no external change coming from
As your
outside
time
the
is
cell
up,
in the Ca2 plus.
IIT Bombay, you have 2 minutes to present your answer. And your time starts now.
So for the fourth question, because the beta's conduction time is less, so majorly the conduction will happen faster in beta, and its recovery time is greater than alpha. But also for it to conduct in beta, the recovery time of beta should be less than the conduction time of beta. So that is one condition which should be there. And also in case beta is in the recovery phase, then the impulse will conduct from the alpha channel, and it may not completely alternate as alpha beta, but whenever beta is unable to pass an impulse through it because of it being in a recovery phase, the alpha channel would allow it to be conducting, and hence allowing for the normal function. Yeah, it will ultimately form a periodic function because the heart impulse will also be a periodic function. And in case your electric signal is incorporated such that both alpha and beta are in its respective recovery phases, then that signal won't be able to pass through it.
So in the first question, what is the role of somekisome? So it is basically binding to the calcium ions on the receptors. So because of which the concentration in the cell is in the cytosol is decreasing, because of which we can see sparks and the frequency is increasing because of that. And why do we see calcium sparks in unstimulated cardiac cells? So what happens is
IITB, your time is up. The floor is now open for 2 questions from judges. No questions. We now request a member of the question making team to come up to stage and present the correct answer.
The final question that is remaining is the memory remains. This is directed to IISER Kolkata. Naive T cells have a unique ability to give rise to long-lived T cells called memory T cells, which can remember the immune response required against a particular pathogen which had infected the body and provide significant immunity against reinfections. Effector T cells are the ones which actually clear the pathogen-infected cells during the infection. T cells have receptors which bind to viral proteins and thus get stimulated. Through a process called asymmetric proliferation and differentiation, stimulated naive T cells give rise to effector and memory T cells. In a genetically modified mouse in which all the naive T cells have an exactly identical receptor that recognizes a specific protein expressed by the FTB virus and binds to it with low affinity, SID injects the FTB virus and tracks the development of effector cells and memory cells over time. A similar experiment was conducted with a mouse expressing high affinity effector T cell receptor, and the given trends below were observed. The top graph is for a low affinity system and the bottom graph is for the high affinity system. Question 1. Make a diagrammatic model of the trajectories of T cell proliferation and differentiation that together explains the above mentioned graphs. In another genetically modified mouse with no T cells, SID added low affinity and high affinity T cells in equal proportions from the mice mentioned above and then repeated the same experiment as above. The graph for his observation is given below. Question 2. Based on your model, explain the above graph. Sid was curious to examine how this model works in the case of an infection with a virus, FTBQ, that replicates much faster than the FTB virus mentioned above. FTB and FTBQ are exactly identical except for their replication rates. Sid injected FTBQ in the high affinity receptor bearing mouse. The experiment results are presented in graph 1 below. Interestingly, Sid observed the emergence of a different population of T cells, which was distinct from either naive or effector T cells. He called them precursor T cells. A protein named RUPSHA is present only in naive T cells and gets activated when the T cells are activated. Sid took normal mice or mice deficient in the RUPSHA protein, both expressing the high affinity receptor for virus, infected them with FTBQ and followed the emergence of effector and precursor T cells. He also measured the virus antigen load in the mice at the various time points. The graphs below show the findings. Question 3. What is the possible role of RUPSHA? Question 4. What is the cause of the decline in the T effector cells seen in both graphs? Question 5. How are the effector and precursor T cell populations maintained in region Z? Question 6. Explain the difference in viral load seen in the three graphs with time. Why do effector T cells from points 1, 2 and 3 show different effects on the viral load? A clarification, RUPSHA activation is observed only in FTBQ infection. IISER Kolkata, do you need any clarifications? Okay. Teams, your thinking time starts now. Just to repeat the clarification, RUPSHA activation is observed only in FTBQ infection. Teams, you have 5 minutes left. Teams, you have 2 minutes left. Teams, you have 1 minute left. Teams, your time is up. IISER Kolkata, kindly come up to the stage and present your answer. You have 5 minutes and your time starts now.
So, so basically, this can be a very rough diagram of a T cell receptor. So basically, when an antigen binds, whether this is a strong antigen, this is a strong binding or weak binding, is mediated through an downstream signaling cascade. So as we can see that the development of memory cell is different in both the cases. So the strong binding and weak binding of the antigen is conveyed differently. But we can say that the effector cells develop similarly. So after the T cell receptor is stimulated by the antigen, we have development of effector cells, whether it is a strong antigen or a weak antigen. But when a strong antigen binds, somehow there's another signaling arm which leads to development of memory cells. Or the T cells start secreting factors which lead to development of other T cells around it into memory cells. So this is our model to explain these two graphs, where as a strong antigen binds, both the arms of this pathway are activated, where effector cells and memory cells both are being developed. But there's also a third thing which ensures that after some time, when the antigen starts to get cleared, somehow the pathway of the activation of helper cells is activated, so that they can stay in the system to clear out any infection which happens in the future. So the second graph is to explain the second graph. It can be thought of like that. First, let me answer question number third. So what is the role of RUPSA? So RUPSA can be thought of a protein which is required for like differentiation of the native T cell to the precursor T cell. Without it, we see that there is no development of precursor T cell. And the precursor T cell can be thought of a regulatory element which somehow is regulating the function of effector T cell. And it is not seen in the case where the viral load was less or the virus was less replicative, because then we didn't require so there is a chance that the immune system could be hyperreactive due to high viral load and can lead to severe inflammation. So the body leads to the body develops precursor T cells which check over these effector T cells, and later on this is asking that what is the cause of decline in T cell effector cells in both the graphs. So as the cells are activated, they are programmed to die after some time, so that they won't stay in the system for too long and cause too much of inflammation. So that can be said to be the case of decline. And how is the decline not happening in case of the place where effector cells are there? So effector cells can be thought of releasing a don't die signal to the precursor cell, can be thought of releasing a don't die signal to effector T cells, which keep them alive, telling that okay, this time this is a strong pathogen, and this is the case. And to explain the fifth graph, we see that the T cell which is taken from region 1 is most potent. It's most we can say that this T cell has the highest potential to release inflammatory molecules and cytokines. So this is the case. And 2 and 3. So for 2 and 3, we can say that 3 is more effective, because after the effector cells leads to see what happens is that okay. So during the course of infection, these T cells grow old and lose some of their cytokine.
30 seconds left.
These T cells grow old and lose some of their cytokine ability, but these somehow the regulator cells are rejuvenating or giving an proliferative signal to these T cells to secrete. So we can see that the here 3 is more potent at 2, and 1, since these are the newest T cells or the most actively secreting T cells, they show the highest response against the virus.
Your time is up. IISc, you can now present your answer. You have 3 minutes and your time starts now.
Okay, so I will begin with the mechanism that we think works on how the cells are proliferating into the effector and memory cells respectively. The difference in this case is that E cells require a certain threshold of the antigen naive cells require a certain level of antigen to convert to E cells. Now in the first case, when there was a low affinity of the binder of naive cells to the antigen, the receptors are quickly saturated, and that leads to E cells forming memory cells, whereas in the second case, due to high affinity, it can continue detecting the no antigen is sort of misleading. What I mean is it is unable to detect any more antigen. It needs to detect the antigen continuously. If it isn't able to do that because of saturation, which happens in the first case, it becomes memory cells, whereas in the second case, it only stops detecting the antigens once it has cleared out the viral infection, which is why we only see an increase towards the end of the graph, increase of memory cells towards the end of the graph, and E cells are dying as the infection gets cleared out. For the third graph, it can be explained on the basis of affinity again, where high affinity of E cells causes an initial increase in E cells only of the high affinity cells, and after a point when we have enough viral load, even the low affinity cells start proliferating, leading to a kink in the graph, and at the same point we see an increase in the memory cells, the same mechanism that was happening in the first initial graph. About the second part of the question, RUPSA is a protein that stimulates the conversion of native cells into the precursor cells, and precursor cells have the ability to turn into effector cells only when the effector cells undergo wear and tear, which we see in the graphs. The decrease of T effector cells is because of wear and tear, because of constantly fighting the infection, and for a while they are able to control the viral load, but once they start declining, the viral load
30
increases
seconds
again.
left.
This causes the P cells to turn into E cells, and the E cells around the point 2, the maintenance of E cells is because of P cells turning into E cells, whereas in the second graph, E cells collapse because there are no P cells to continue building E cells. What did I miss? Yeah, that's about it. And yeah, to explain the last graph, one has the strong...
Your time is up. IIT Bombay, you can now present your answer. You have 2 minutes and your time starts now.
So basically, for the first two questions, we have this model which is getting followed. The naive cells are forming if there is low affinity of the effector the antigen, it forms memory cells and the effector cells. Whereas if it is high antigen binding capacity, it forms only the effector cells initially, and after the antigen has been wiped out, it starts forming the memory cells. The RUPSA is the protein which gets formed and is responsible for the formation of the precursor T cells. And the decline is seen because T cells are short-lived cells which are only responsible for fighting the infection, and after that has been done, they die. They have a very shorter lifespan compared to the memory cells. And the effector and precursor T cell population is maintained in region Z. That's because the precursor cell is responsible for the for the formation of effector cells again. So even though the effector cells start dying after a particular point of time, they are maintaining a constant concentration because of the precursor cells. And if we see, even the virus load or the antigen load decreases after the third point, after which the effector cells are able to fight the antigen. And in the last graph, if we see the T cells taken from the first set of the point 1 is more potent because it is directly from the naive T
30
cells and
seconds left.
they're effective in fighting the antigen. The second point is where the T cells have been formed from the precursor cells and they have not been bound to the antigen, because of which they're not able to so effectively fight against the pathogen. And in the third point, they have been there for a certain amount of time, because of which they have the memory and they're able to fight, and the potency is more than the second but lesser than the first.
IIT Delhi, you can now present your answer. You have 2 minutes. Your time starts now.
If there's the high affinity T cells, they would attach to all the antigens, and they would first kill all of them, and then they would start forming memory T cells. And if for the low affinity one, since they have low affinity, some of them will attach to the some of them would attach, and after it has killed the antigen, then they would start forming memory cells, because they would have very low affinity for other antigens. And so initially both of the, initially as the T cells attach to the antigens, it starts forming the effector T cells, and simultaneously with a very small delay, memory T cells are formed. And then after a certain time, an equilibrium has achieved when the number of T cells binded and the number of T cells which has been already killed the antigens have been equal. And when the effective T cells and they both are equal in number, initially the high affinity T cells will attach to the antigen, and so till a certain time, and
30
then
seconds left.
and then the low affinity T cells will start to kill the antigen, and then again a equilibrium will reach, and so the same.
Mimamsa is a protein that affects the process of differentiation, activating a pathway in the differentiation process to form precursor cells. And I believe the effector and precursor T cells populations are maintained because the precursor T cells has a secondary function where it also attacks.
Your time is up. The floor is now open for questions from the judges. We request a member from the question making team to present the correct answer.
How do I pass this?
Audible, right? Okay. Yeah.
You just go. I don't need it. So good question. Good answers, team. I think all of you gave partly correct answers to some parts. So I'll just start explaining everything, not just part by part. So these graphs, as correctly shown by IIT Bombay team, in the low affinity model, initially both effector and memory populations arise, and after a point when antigen is cleared, some effector cells die and some are converted into the memory cells. This is different in the high affinity case, in which all the effectors, all the naive cells are differentiated to an effector fate, and when the effector cells are dying, similarly in this case, they give rise to the memory cells. Physiologically, this is relevant and also somewhat rational, because it is better if you direct the high affinity clones to an effector state, because they would be more equipped to kill the antigen infected cells. And this, I think IISc team gave a rather right answer for this, that initially high affinity clones outcompete the low affinity clones, and once all the high affinity clones are converted to an effector state, then the low affinity clones start proliferating, and they give rise to effector and memory population both, and similarly when the cells die, they give rise to the memory cells. So what Rupsha protein is doing was also correctly answered by almost all of you, that it detects not just the affinity is detected by the receptor, but Rupsha protein detects the amount. So it is a sensor of amount of viral or infection load, and then it directs the differentiation of an intermediate state called progenitor cells. And these progenitor cells, so essentially what happens is that after these cells die, effector cells in, this case, for example, in the chronic infection case. So now, also, answers for that, all answers are acceptable. It was rather an open-ended question. Inherent lifespan of the cells is an acceptable answer. I think IISER Kolkata team answered that they are programmed to die to avoid self-inflammation and severe inflammation. That is also a correct, acceptable answer. And after that, what happens is the source of effector cells shifts from naive population to the precursor population. And these precursor population give rise to cells which are inherently not as cytotoxic or primed to function as the initial effector population. That also explains the initial graph or the last three graphs in which the cells taken from the early effector population clears the antigen or viral load the most efficiently. But the paradox arises in the second and third case. If I'm taking cells from the last point, then why are they clearing it better than the ones you're taking from the second point? So what we wanted you to come up with was that these cells, the ones that are coming out of the precursor state, actually undergo a linear differentiation trajectory in which they go from a lower cytotoxic function to the higher cytotoxic function. Kolkata answered that precursor cells can give signals to these effector cells so that they can sustain, but then it will not very nicely explain why, if I take effector cells out of one mouse and subject them to another mouse which does not have the precursor population, why does that clear the viral load so well? So overall, yes, congratulations on your answer, and that's it. Thank you.
With that, we conclude the deep thought round for biology Mimamsa. Thank you to everyone for attending.
First of all, before we all proceed outside, I want to just formally thank everyone here. We once again extend our gratitude to the judges, Professor Satyajit Rath and Dr. Sarita Puri, all the volunteers and all the coordinators. Also, we will be starting the maths deep thought at 5:40. And in a while, we'll be proceeding outside for snacks. So all the volunteers, all the participants, judges, will be. You can all of you can join us for snacks outside. We'll be starting them shortly. Thank you so much.